I haven't even left yet and I'm already missing you guys. All right, we are going back to our Doodle notes. So if you don't have those out, go ahead and get those out. Just pause this and then resume when you are ready. And then you should have also collected the LT6.3 practice assignment that's on the black cart. As you're taking notes today, be sure to pause where needed so that you can get the information down in your packet. All right, today our focus is right here on LT6.3. We did our blood typing lab, which you should have turned in today. And now we're going to look at how those blood type alleles are inherited. So we're going to use Punnett squares and the rules of probability to make predictions about the genotypes and phenotypes of genetic crosses that involve multiple alleles, and we're going to focus on blood types. So get this information down in your notes, but remember that we've already talked about complete dominance, which is where you have the same letter, you're using the same letter to represent the allele, but you have a dominant allele and a recessive allele. So we're going to see big letters and little letters. And then we moved into incomplete and codominant patterns of inheritance, and that is where we use two different letters and we saw a third phenotype appear. Well, now we are going to discuss multiple alleles. So we're looking at traits that involve more than two alleles for a gene. Now we are still going to only inherit two alleles for our gene. Okay, so our mom is going to donate one and our dad is going to donate one. But there are more than just those two available in the population. So one example of multiple alleles is looking at coat color in rabbits. We see that in rabbits, coat color comes in 4 alleles, and I'm showing those here. All right, and each of those alleles then is coding for a different specificity for the trait. So for rabbits, their coat color comes in brown, black, chinchilla, Himalayan, and albino. So we already know that in humans there are 3 alleles for blood type, and we looked at these alleles in our lab last class. And we saw that we have allele A, B, and O. We also discussed that blood types actually are inherited through 2 different pathways of inheritance or patterns of inheritance. We're going to see that complete dominance and codominance both play a part in the inheritance of blood type. So we know that A and B are codominant to one another, but they're both dominant to the O allele, which is the recessive allele. So a person with the genotype 1 big A, 1 big A or 1 big A little I will have type A blood. All right, and we discussed the 4 different types of blood in our last class. One thing you might want to do is go ahead and put some of that information from last class here off to the side so that as you are going through these genetics problems, you're always referring to them and then you start to memorize them. Because unlike the other genetics problems that we've practiced where the letters did not matter, with blood types, letters absolutely matter. So we have to use the same genotypes at all times when we are discussing blood types. So I'm just going to write it up at the top. But remember, with type A, we could have seen the genotype with the homozygous form of A. And we could also get type A if we had the heterozygous form for A. Same with B. So with B, we could have homozygous B, or we could have heterozygous B. Remember, we see that A and B are codominant, so sometimes we're going to have individuals that have the AB type of blood, and we would show it like that in our genotype. And then the last phenotype is O. And recall that O is the recessive allele, and so we're only going to see O in its homozygous recessive form. So make sure you get that somewhere on this page just for quick reference. But anytime you see a genetics problems that is dealing with blood types, you have to use one of these genotypes to solve it. So we have our first sample problem. We have Aladdin and Jasmine are finally having a baby. Woo woo! As we read through the problem, we are going to be sure to identify important information. So we see that Aladdin has homozygous type A. My pen just stopped working. Give me a quick second. Okay, we have homozygous type A. And then Jasmine has heterozygous type B blood. So then we're going to use the Punnett square like we have been to determine the possible blood types of their child. So heterozygous, remember we're going to have the two different alleles, but it's type B. So we know we have to have a dominant B, and then we're going to have the recessive allele for blood type. And then with Aladdin, he had homozygous type A, so both of his alleles are going to be dominant for A. All right, so then when we do blood typing with genetic crosses, we're going to have to always put the A first because we have AB. It's not BA, it's AB. So we want to make sure that we write these where if we have the letters A and B both, we need to make sure that the A is first. And then always write those dominant alleles first. So since Aladdin had type A, homozygous, I'm going to go ahead and drop his alleles down, and then we'll bring in Jasmine's, and here we have our results. So we ask ourselves, what percent is type O? Oh, by the way, you probably will not see any multiple allele questions that are going to ask you about like genotypic frequency or genotypic ratios or phenotypic ratios and stuff. It'll almost always be percents of the possible blood types. All right, so we don't see any O's because to see O, I would have had to have had the recessive form. A's, we do have 50% A, and we're seeing that in its heterozygous form. But remember, we could have also seen it as the homozygous, potentially. And then B's, we do not have any. And then for our ABs, we have 50%. Okay, you guys, I want you to pause the video, and I want you to work this pre-practice problem out on your own. Then resume when you're done to check your work. Okay, assuming you are ready to move on, we're using a Punnett square to determine the possible blood types of the offspring when the father is type AB. So dad is AB, and the mom is heterozygous for type A. So that tells us that we're going to have this genotype. We're going to go ahead and do the crosses, and I'm just going to solve this. Remember, A goes first. All right, now looking at the results of our Punnett square, we see there are 0% that is O. Type A, we do have 50% because we're seeing type A here and here. And then type B, we have 25%. And type AB is 25%. I hope that's what you got, but just in case, we're going to practice a couple more and work through those to make sure that you are confident in your ability to be successful on our practice assignment. So let's go into our next next pre-practice problem and pause again to do the work yourself. And then when you're ready, resume the video so that you can check your work. This one is a little bit more challenging, so have fun with this one. All right, my assumption is that you are ready to go. So we see that George has type A. That is all we know. I mean, we know that type A has 2 possible genotypes, but we are not told which one it is. All we know for sure is that he has at least one of those. We see that George's mom was type A as well. So again, that's the only information I've been provided. So we know George's mom has at least one A allele. And then his dad is type O. So in order to have type O is the recessive form. So we know what George's dad's genotype is, which is great because now we know that George has to give one of these, or George's dad, that is, has to give one of those to George, and we now know what George's blood type alleles are. All right, so now that we have George, he married Susie, who is type AB. Type AB is pretty easy. That's good. And now we're going to combine these alleles. By the way, this process right here that we're working out is actually the fertilization process that we discussed a few units ago. So during meiosis, remember we made the gametes. That's where we have isolated the alleles. And then during fertilization, that's where we merge mom and dad's alleles to give the potential offspring genotypes here. All right, so here is the results, and then we have to identify the phenotypes. So we see that we have some type A's, because we're going to have type A here, and we're going to have type A here. And then we have some type ABs right here, and we have type B. So there we have that genetics problem solved, and we're ready for our last pre-practice problem. So again, pause the video so that you can do the work on your own first or with your table partner. And then when you are ready, check your work. And we're back for the last check of our pre-practice problems for 6.3. Before we get started here, this problem is like a paternity problem. And so far, like in the lab, what we were using blood typing for was to determine who would be a safe donor for a patient. And so now we're seeing that blood types are used for paternity. Blood types are also used in forensic evidence, but in these cases, like forensics and paternity issues, what we need to realize is that using blood types cannot prove that someone is the father of a child or that someone is the guilty person. We actually have to have more tests to determine that. What blood typing does for paternity and forensic science is that it eliminates possible suspects and possible fathers or mothers. So it's an elimination. It helps us to eliminate. All right, so anyway, back to our problem. So it's 1968. DNA fingerprinting has not been discovered yet, or developed, I should say. And so we're going to use blood typing to try to help these parents identify if their baby was switched at the hospital. So we have a mom with blood type O, so we know that she is homozygous recessive for that blood type. And then dad has blood type AB, and so we know his genotype here. We're going to cross these. We're doing the work. Okay, here we have our results. And so we see, based on the parents that we were provided, that we could have a baby with a type A or a baby with type B. And the baby in question has type B. So was the baby switched? Well, we're not 100% sure, but probably not. And we say probably because, again, we're not 100% sure, but we're saying not because there is a possibility that these could be this baby's parents. All right. So you guys, thank you so much for joining me in this video lecture. It is now time to practice what we've learned and spend the rest of class working on LT6.3 and then turning that in before you leave. That is our goal. So you have no homework. And then next class, we are going to start on 6.4, which is taking a look at pedigree charts, which is like a family tree, but what we're looking at through the family tree is a trait that has been passed from generation to generation. And then we're also going to be looking at karyotypes in this standard. And I know that you guys have done some karyotyping work with your Project Lead the Way class. So have a wonderful, stupendous, fantabulous, superb, awesome, and amazing weekend. I am out.